Very good evening to all the NSEJS aspirants. So here we are to discuss NSEJS 2022 paper. In that, amongst that, 3 concepts we are going to exercise. One from glycolysis pathway, that is in respiration, and 2 are from applied biology concepts. Without much delay, let's get started with the first question. So here if you look into, in the analysis of wastewater, Escherichia coli, that is E. coli, is used as what? In analysis of wastewater, E. coli is used in what ways? That is the question. A standard organism for performing a plate count? No, E. coli is not used as a standard organism for culture plate count. It is an indicator of fecal contamination of water. This is correct because E. coli is a coliform bacteria. E. coli is a coliform bacteria which lies in the intestines of many organisms and fecal matter as well, including in human beings. It lies in the intestine. E. coli, which is a gram-negative bacteria, coliform bacteria, which stays in the intestines of human beings, and this is a very good indicator of fecal contamination of water and an indicator of the number of nitrogen fixing bacteria. E. coli has no relation with nitrogen fixation, so you can eliminate this option as well. It is a measure of amino acid content of water. No, E. coli has no role in amino acids or protein content in the water. So this is used as what? An indicator of fecal contamination of water. Look into the theory also, beta. E. coli or Escherichia coli is a coliform bacteria. Okay, this is a coliform bacteria that lives in the intestines of warm-blooded animals, including humans. Fine, and it is a non-pathogenic. It is non-pathogenic in most of the cases, but its presence in water will increase the toxicity of the water. Okay, its presence in the water will increase the toxicity of water, and that leads to what? Water contamination. And next, if you see, this makes E. coli as what? Biological indicator of water quality. So this is a biological indicator as well, and pathogenic bacteria like Salmonella and Shigella are most dangerous but harder to test directly. Hence, E. coli can be used as a proxy, okay, than detecting for harmful bacteria like Salmonella and Shigella. We can use E. coli as a detector for water contamination because it is easy to test it directly. And for standard plate count methods, this will measure total microbial load but not specifically fecal contamination. So, with E. coli we cannot conduct the standard plate count. And then E. coli is not a nitrogen fixer. So, it is wrong. E. coli is not a nitrogen fixer and it has no role in amino acid quantification. It has no role in the amino acid quantification. So, E. coli is purely involved in what? It is purely used as an indicator of fecal matter of water. And then if you see, beta, what are the best references to learn about more bacteria, be it harmful bacteria or useful bacteria like E. coli, which is gram-negative bacillus coliform bacteria, right? And then here we have got a question on the nitrogen fixing bacteria also, where we have many number of symbiotic bacteria and free living nitrogen fixing bacteria as well. To know the complete role of bacteria in the applied biology, what are the best books you have to refer? NCERT class 11, of course, biology. From unit 5 human physiology, you can go through the microbial world in that, and then from digestion and absorption and microbes in human welfare, you will get complete picture on the bacteria, both useful and harmful bacteria. And then from Truman's elementary biology of part 2, where you will get complete reference of bacteria with the objective from part 2 applied biology. And if you have to go through the standard reference books, you can also go through Campbell Biology, which is the most standard book where you can go through the sections of water pollution and indicator organisms, and environmental microbiology section. These are the best reference books where you have to go through beta. And now second question, it is very basic, okay. Acid rain damages soil. Acid rain will decrease the pH, right ma? Acid rain will decrease the pH of the water. As such, it will damage the soil and lakes, which pH range is most suitable for the survival of aquatic biota. Which pH range is more suitable? That is the question. Which pH range will be most suitable ma? Neutral pH should be there. Neutral pH. Both for the soil and aquatic organisms like lakes should be healthy, so the pH range should be what? Neutral pH. Amongst these four options, which is the neutral pH? 7.5 to 8.5, so this is basic. This is acidic from 4.5 to 5.8. 6.5 to 7.5 is neutral pH. Okay. Now you look into the explanation as well, theory as well. So here if you see ma, most aquatic organisms like fishes, amphibians and planktons require a near neutral pH for their survival, and a range of 6.5 to 7.5 pH will provide stability for enzymes, respiration and osmoregulation. Acid rain includes sulfates and nitrate gases which will dissolve the rainwater, and they will form what? Sulfuric acid and nitric acid. Sulfuric acid and nitric acid when they are mixed with water. As such, the pH gets reduced, and as it is involving the acidic solutions inside it, we call it as acid rain, which is highly damageable for soil and lakes, aquatic life. It is very dangerous for the aquatic life and for soil also. This makes it unsustainable for aquatic life. If pH falls below 5.8, most fish species like trout cannot reproduce. So this is one point to be noted with the example. And high alkalinity above 9.0, this will also damage especially gills and disrupt the physiology of the aquatic organisms. Fine, especially fishes. Hence, a balanced range of pH is around 6.5 to 7.5, which is most essential. Clear, right ma? Now, what are the best reference books for this basic concept? From NCERT 11, you can go through the environmental pollutions chapter, which is unit 10, and you can also go through organisms, populations and environmental problems, and you can get a best reference from Arihant NSEJS guide, ecology and environment section, and Odum's ecology, which is the standard book where from this concept you will get complete clearance on the acid rains and the aquatic ecosystems, basic concepts of aquatic ecosystems and acid rain effects. From this you will get the complete picture of the pH, range, aquatic life, acidic rains, and what should be the range to maintain the aquatic life properly. So with this, 2 questions are done. Third question is from physiology, which is very important. Fine, and the question is from glycolysis, which is also called as EMP pathway. Emden, Meyerhoff and Parnas. Okay, named after these 3 scientists, and it is also called as EMP pathway, as this is a common mode of respiration or common pathway which is operated both in anaerobic respiration and aerobic respiration. This glycolysis is also called as incomplete oxidation or partial oxidation that takes place. In glycolysis, incomplete oxidation or partial oxidation of glucose occurs. So 2 points to be noted, all students please take down ma. This is operated both in aerobic and anaerobic respiration, both in aerobic and anaerobic respiration. Fine, it is operated both in aerobic and anaerobic respiration. And what type of oxidation happens in glycolysis? Glucose is incompletely oxidized, or you can also say partially oxidized. Fine, right? So look at here carefully. In the following EMP pathway, enzymes catalyzing the reactions are numbered from E1 to E10, from enzyme 1 to enzyme 10. At which enzyme levels ATP are generated at substrate level? So he is asking you at which steps SLP is happening, substrate level phosphorylation. So now I will be telling you not just about SLPs, I will also tell you about the irreversible steps, which step is the rate limiting step, and we will also discuss about the all enzymes that are included in glycolysis pathway. Look at here carefully, ma. At the very first step, that is glucose is being break down to glucose 6-phosphate by the enzyme hexokinase. By what enzyme, ma? Hexokinase. Fine. Glucose 6-phosphate to fructose 6-phosphate. This is by phosphohexoisomerase. Fine. Now, fructose 6-phosphate to fructose 1,6-bisphosphate. This is the third step, which is called as rate limiting step. Third step is called as what, ma? Rate limiting step, and this is catalyzed by phosphofructokinase. So here you make a note of it, ma. At first step and third step, ATP is being utilized from the cytoplasm. Okay, ATP is being utilized for phosphorylation step here, both at first step and third step. Okay, and at how much speed the glycolysis have to take place? The accuracy of glycolysis is maintained by third step of glycolysis. That is why third step is called as what, ma? Rate limiting step, and phosphofructokinase is also called as phase maker enzyme. This is called as phase maker enzyme. Fine. Now, fructose 1,6-bisphosphate, it will cleave. Cleavage happens at this step, wherein the 6-carbon compound is cleaved to 2 molecules of 3-carbon compound, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and this step is catalyzed by the enzyme aldolase. Okay, and now it is 6th step beta, DHAP or G3P2 phosphoglyceraldehyde by isomerase enzyme, phosphoglycerate isomerase enzyme, and 3-glyceride phosphate or 3-phosphoglycerate to 1,3-bisphosphoglycerate. This is by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Fine, that is E6. Now, glyceraldehyde 1,3-bisphosphoglyceric acid to 3-phosphoglyceric acid. This is catalyzed by the enzyme phosphoglycerate kinase. Okay, and 3-phosphoglycerate to 2-phosphoglycerate by the enzyme phosphoglycerate mutase. 2-phosphoglycerate to phosphoenol pyruvate by the enzyme enolase. Lastly, from PEP to pyruvic acid, the last enzyme is pyruvate kinase. Clear, right? Understood all the enzymes? First it is hexokinase, from glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate by phosphohexoisomerase, and fructose 6-phosphate to 1,3-bisphosphoglyceric acid by the enzyme phosphofructokinase. First step and third step are irreversible steps, and third step is called as what, beta? Rate limiting step, as this step will decide the accuracy of the entire process. And then fructose 1,6-bisphosphate 2, here at the cleavage step, it will be cleaved to 2 molecules of 3 carbon compound, that is DHAP and glyceraldehyde 3 phosphate. So that is catalyzed by the enzyme aldolase, and here DHAP to 3 phosphoglyceric acid by the enzyme phosphoglycerate isomerase, and 3 phosphoglyceride to 1,3-bisphosphoglyceride by the enzyme glyceraldehyde 3 phosphate dehydrogenase, okay, and 1,3-bisphosphoglyceride. So at this step wherein 3 phosphoglyceride to 1,3-bisphosphoglyceric acid, here it is dehydration step where it will be releasing out 2 molecules of NADH from one glycolysis. So from one glucose molecule if you have to take, at this step 2 NADH will be released out. And at 7th step from glyceraldehyde 3 phosphate, that is 3 phosphoglycerate to 2 phosphoglycerate, one more here 1,3-bisphosphoglycerate to 3 phosphoglycerate, here one more ATP will be released, okay. If you have to consider for one glucose molecule, from 5th step, as 3 carbon compounds are there, right, 2 cycles will happen. So you have to count 2 NADH and 2 ATP, clear, right ma? So now here at 7th step 2 ATP will be released, and at 10th step also 2 ATP will be released. So this ADP and inorganic phosphate will be taken from the substrates, okay. Inorganic phosphate for the formation of ATP are being taken from the substrates. So these 2 steps, that is 7th step and 10th step of glycolysis, are said to be substrate level phosphorylation, wherein ATP formation, for ATP formation phosphate group, that is inorganic phosphate, is being taken from the substrates. So 7th step and 10th step are called as what? Substrate level phosphorylations. So here you make a note of it ma, we have 2 SLPs, 2 substrate level phosphorylations, one at the 7th step and one at the 10th step, okay, and we have 3 irreversible steps. 3 irreversible steps. Irreversible steps include 1st step, 3rd step and 10th step. These are the 3 irreversible steps. So in question, as in question, he has asked us about substrate level phosphorylation. Our answer should be option C, because it is enzyme 7 and enzyme 10 that are being acting on what? Substrate level phosphorylation. So here you can see substrate level phosphorylation means what? ATP is made directly by transferring a phosphate group from high energy substrate to ADP. Inorganic phosphate is transferred from high energy substrate to ADP. Like in 7th step, if you have to take, it is being transferred from 1,3-bisphosphoglycerate, and in 10th step, if you have to take, the inorganic phosphate is being transferred from phosphoenol pyruvate. As such, wherein the energy is being transferring a phosphate group from high energy substrate to ADP for the formation of ATP. This is called as SLP. This happens at 2 steps, one at 7th step and one at 10th step. So here once again, if you have to look into theory, 1,3-bisphosphoglycerate to 3-phosphoglycerate, where high energy phosphate of 1,3-bisphosphoglycerate is transferred to ADP and 1 ATP is formed. 2 ATP is per glucose molecule, because from 5th step 2 turns of the cycle will happen. Next, pyruvate kinase. Here if you see ma, pyruvate kinase, phosphoenol pyruvate to pyruvate. PEP has very high phosphoryl transfer potential, wherein phosphate is given to ADP for the formation of ATP. Okay. So here 1 ATP per triose is there, as such 2 ATP per glucose are formed. And here if you see mapping of enzymes, all the enzymes which we have discussed are there over here beta. If you want, you can take a screenshot of it. And lastly, what will be the best reference books that you have to refer for this physiological concepts? Now only the concept is asked on glycolysis. Glycolysis, which is operated in all prokaryotes, eukaryotes, anaerobic respiration, aerobic respiration. Glycolysis happens in cytoplasm of a cell, right? So, the question can be put on any of the part of respiration. It can be from fermentation if anaerobic respiration is considered. They can ask a question on lactic acid fermentation or alcoholic fermentation, or if aerobic respiration is concerned, they can ask a question on they can ask a question on oxidative decarboxylation or tricarboxylic acid cycle, or the question can also be put on electron transport system. So, to cover up all these physiological pathways, which books we have to refer? What are the best books to cover up the physiological concepts? Of course, from NCERT Biology class 11, best reference book is what? Respiration in plants. From NCERT class 11, respiration in plants, glycolysis section you have to go through. And from Arihant, NSEJS IOQ JS Junior Science Guide, Biology, Cellular Respiration and Metabolism should be taken. And then standard reference books include Campbell and Leninger beta, where in this what sections you have to refer is cellular respiration and fermentation from Campbell. From Leninger book you have to go through glycolysis, gluconeogenesis, which means that synthesis of glucose from non-carbohydrate sources, that is gluconeogenesis, and pentose phosphate pathway. These are the sections you have to go through from the standard reference books, that is Campbell and Leninger. Clear, right beta? So, with this we have finished out NSEJS 2022 major questions from biology. In our next session, we will be looking into the questions of NSEJS 2021. So, stay tuned, and if you want to know about all the concepts so far we have discussed, you can go through the entire playlist beta. So, that is all for today's session. Thank you.